Hello, my name is Kira Matthews, and the title of my research is Single-Cell RNA Sequencing Reveals Differences in Myeloid Cell Transcriptome Enriched in the Inflamed Intestines of Ulcerative Colitis Patients. So ulcerative colitis is an autoimmune condition whose pathogenesis is largely unknown. I sought to develop a greater understanding of this pathogenesis by investigating myeloid cells. So ulcerative colitis, or UC, is characterized by chronic inflammation, and the pathogenesis is largely unknown, although it has been identified as an immune-mediated condition. So the question remains, what immune mechanisms are driving ulcerative colitis? Bao et al. 2020 found that myeloid cells have been implicated in UC pathogenesis. Thus, I chose to investigate three subsets of myeloid cells, the first being macrophages, which are unique in that they are responsible for the largest amount of phagocytic activity within the gut. However, there is a need for further research investigating the functional heterogeneity of phagocytes, such as macrophages and other myeloid cells, within UC. Secondly, I chose to investigate monocytes, of which there are three subsets: classical, intermediate, and non-classical. However, there's a lack of research investigating the functional heterogeneity of monocyte subsets within ulcerative colitis. Thirdly, I chose to investigate dendritic cells, of which there are two subsets: MDCs and PDCs. However, the functional heterogeneity of these DC subsets is unknown within UC. So the broader aim of my research was to develop a greater understanding of which immune mechanisms are causing the ulcerative colitis and contributing to that inflammation. So within the context of UC, this study aimed to elucidate the functional heterogeneity of macrophages, monocytes, and DCs, and I hypothesized that the following cells play a dominant role in UC: macrophages over all myeloid cells, classical monocytes over those monocyte subsets, and MDCs over DC subsets. So my mentor provided me with single-cell RNA sequencing-derived gene lists, and from these gene lists, 25 gene clusters were produced. From these clusters, I isolated those clusters which were enriched in myeloid cells to continue my investigation into myeloid cells, and additionally I performed disease enrichment analysis to ensure that these clusters could be used to draw conclusions about UC pathogenesis. Additionally, I used the database Human Protein Atlas, and this is novel in that, to my knowledge, this database has never been used to draw conclusions about myeloid cells within ulcerative colitis. And additionally, the advantage of using this database is that the normalized expression values for these genes were used, which provides broader insight into the pathogenesis of UC. So all statistical analysis was performed on GraphPad Prism 8 with a statistical significance held at p is less than 0.05. Enrichment analysis was performed on Enricher, and the myeloid cell functional heterogeneity was elucidated using one-way ANOVA and two-tailed t-test. So my first step was, from these 25 clusters, which clusters could be used for my investigation, and I found that cluster 9 and cluster 21 were enriched in myeloid cells. Thus, these clusters were isolated for further investigation. I also wanted to ensure that these clusters could be used to draw conclusions about ulcerative colitis, and I found that both of these clusters were enriched in UC and UC-associated inflammation. However, cluster 21 did display greater combined score than cluster 9. Thus, cluster 21 is considered slightly more indicative of UC pathogenesis. To further confirm that these two clusters could be used for my investigation, I performed pathway analysis, and inflammatory pathways associated with ulcerative colitis were enriched in both of these two clusters, which indicates that both of these two clusters can be used to draw conclusions about UC pathogenesis. So beginning the investigation into the functional heterogeneity of myeloid cells, I compared the expression pathways of these different myeloid cells, and I found that the greatest frequency of unique expression was found for macrophages, which indicates that macrophages are playing a greater role in UC pathogenesis when compared with other myeloid cells. So if you recall, cluster 9 and cluster 21 is slightly more indicative of UC pathogenesis. So if we were to expect a cell type to be displaying a greater role in UC, then we would anticipate that it have a greater normalized expression value in cluster 21 when compared with cluster 9, which is exactly what we found for macrophages, MDCs, and PDCs, which indicates that macrophages, MDCs, and PDCs are playing a greater role in UC. Additionally, I compared the mean normalized expression associated with these different myeloid cell subsets, and I found that macrophages displayed the greatest normalized expression when compared with other myeloid cells, which indicates that macrophages are playing a greater role in UC when compared with other myeloid cells. Additionally, I wanted to elucidate the functional heterogeneity of monocytes. However, I was unable to find any statistically significant difference in the normalized expression values between the monocyte subsets. So while this did address my purpose, it did not support my hypothesis that classical monocytes would be playing a greater and more dominant role in ulcerative colitis. And finally, I wanted to elucidate dendritic cell functional heterogeneity. And I found that in both clusters, MDCs displayed greater normalized expression when compared with PDCs, and this actually indicates that MDCs are playing a greater role in UC pathogenesis when compared with PDCs. So in summary, my research focused on ulcerative colitis and investigating the functional heterogeneity of myeloid cells using the database Human Protein Atlas and genes derived from our single-cell RNA sequencing. So a limitation of my study might be that only 12 biopsies were collected and gene lists were produced from these biopsies. However, in single-cell RNA sequencing studies such as Liu et al. 2021, a biopsy sample of between 10 and 20 is typical. Additionally, I used the normalized expression values associated with these genes, which allows my research to draw broader implications. Perhaps the most interesting finding was that I found that while MDCs and PDCs both displayed greater normalized expression in cluster 21, MDCs displayed greater normalized expression than PDCs. So while MDCs can be considered a potential therapeutic for ulcerative colitis, further investigation is warranted into the role of PDCs in ulcerative colitis. It's unclear whether it's playing a pathogenic or non-pathogenic role. So I found that macrophages, MDCs, and PDCs displayed greater mean normalized expression in cluster 21 when compared with cluster 9, which indicates that macrophages, MDCs, and PDCs are playing a greater role in UC compared with other myeloid cells. Additionally, I found that macrophages display greater mean normalized expression when compared with other myeloid cells in enriched UC clusters, which indicates that macrophages are actually playing a greater role in the pathogenesis of ulcerative colitis when compared with other myeloid cells. And finally, I found that within the DC subsets, MDCs are displaying a greater mean normalized expression when compared with PDCs in enriched UC, which has great implications for the functional heterogeneity of DC subsets because it indicates that MDCs are playing a greater role in UC pathogenesis when compared with their counterpart PDCs. So the broader contribution of my research is that I have developed a greater understanding of UC pathogenesis and the immune mechanisms which contribute to inflammation in UC, which is critical for the development of more efficacious treatments for ulcerative colitis. I would like to thank my mentors for providing me the opportunity to complete this project, my science research teachers for their support, and my peers for their endless guidance. Thank you for listening.